Okay, well, it is a great pleasure for me to introduce you to somebody that most of you have probably never met before. So I welcome her to our community. This is Dr. Cristina Alberini. You're going to fall in love with a couple of things about her. The first is her accent. That's wonderful. And the second is her unbelievable ability to really talk to families. So Dr. Alberini, a little story about her is that it was maybe 2 years ago now that I may have even been here. And I always check my email because, I have to answer a lot of those. And I think something came off of my email, and it was an inquiry if FAST was interested in funding some interesting work that she happened upon, essentially, and I'm sure she'll tell you her story. And she wanted to know if there was interest in funding further research from some pilot information that she had, and she almost accidentally started working with Angelman syndrome. And she showed me these behavior rescues in these mice, these adult mice, and I dropped to the floor that I couldn't believe that she was able to rescue some things in these mice that nobody has been able to rescue in adult mice before. And so we were unbelievably shocked by her and so excited that we couldn't breathe. And then we obviously went forward and funded her. And she's at a point now where she's ready to show you some pretty significant data. And we are really excited about the promise of an approach that she has brought forward to Angelman syndrome that we never even thought about. So Dr. Alberini is a world-renowned clinician. She's a scientist at NYU in New York City. And that also helped because then she's like, do you want to just come to my lab and I'll show you the data? And then that was fun because I got to meet the mice and stuff. She works at Immunological Sciences. She's from the University of Genoa in Italy, and she trained in neurobiology as a postdoc fellow at Columbia. From 1997 to 2000, she was an assistant professor at Brown and an associate and full professor at Mount Sinai School of Medicine from 2001 to 2011. And she's now full professor in neuroscience at NYU. So I'm very excited to introduce Dr. Alberini as the new member of our team.
Thank you. Thank you so much, Alison, for the wonderful introduction. I'm honored and very happy to be here to share with you the story that we see day by day progressing on Angelman syndrome in the lab. And particularly, I'm going to talk about a small protein known as insulin-like growth factor 2. Small proteins are also called peptides, that we identified at the beginning as a memory enhancer. And in fact, just to give you a little bit of the history of the lab, my lab was not working on neurodevelopmental disorders. We have been working on the biology of memory. What we are interested in is to understand how the brain changes the biology in response to learning, and how this biology is important to form long-term memories and store them. And particularly, we are studying the memories that are called declarative in humans or episodic. These memories are the memories that you can talk about, the memories of who you are, where you have been, who you met, what you did last week, what you did 2 years ago, what is important in your life. And these memories, as you can see in the first slide, are combining are processing all the information about places and people and things, and also the time that binds together all these elements. So these are typically the episodic memories, right? An episode. So you have been here today, you will remember to be here, you will remember more or less details depending on how important this episode was for you. And these memories in humans require the medial temporal lobe. It's a region behind our ears. And one of the regions involved is the hippocampus. And the hippocampus crosstalk to many other regions in the brain to process this information and store long-term memories. And this is true also in other animals, like rodents. The system is very well conserved in the sense of the brain regions involved, as well as the biology underlying the formation and the storage of these memories. So in rodents, we can study the hippocampus and the cortical regions involved in what these animals store about who they encounter, what, where, and when. And these are the classical memories that are processed by the medial temporal lobe. So the questions that are ongoing in the lab is to understand what is the biological response to this type of learning and how these responses store the memories. So we are looking in particularly at gene expression, and in fact, the brain is very plastic. When we encounter some new experience, our brain changes. So your brain is changing right now, and it will change in response to experience. And these changes are characterized by expression of genes. So a number of genes are going to be expressed through this experience. And a number of them are going to be involved in storing the information that becomes long-term memories. So we are interested in identifying what and which those genes are, what they do, how the proteins affect the system, where all these changes happen in the brain, so the different brain regions involved. And we are also interested in understanding the biology of remembering. So once the memories are formed, when we recall them, how does the brain changes? So all these mechanisms are actually characterized by, as I said, cascades of gene expression that underlie what is called brain plasticity. And part of this brain plasticity is synaptic plasticity, which is very much involved in everything we do. So we started by studying this mechanism in the hippocampus. You have heard about this region already from Ed Weber and the plasticity of hippocampus involved in memory. And we are now moving to other regions of the brain. And we have identified, starting from the hippocampus, a number of molecular cascades that are activated by learning. And if we manipulate those cascades, we have no long-term memory formation. So we know those are required for long-term memory. And this gene expression happens in different cell types. It's not only neurons, it's also glia cells, other cell types present in the brain that are less studied still, but now more recently they gain a lot of attention. So by studying all these changes, we identified one that is insulin-like growth factor 2. It's a growth factor which is part of a system known as the IGF insulin system. And in this system, we have 3 proteins: insulin, insulin-like growth factor 1, and insulin-like growth factor 2, or IGF-2. And they bind with different affinity, with different strength, to other proteins present in the surface of the cells called receptors. So IGF-2 binds to another protein on the cells and activates a number of changes in these cells. And the affinity, the strength of this interaction is very important because it tells us what type of changes are going to happen in the cell. And using this IGF-2, we have been studying IGF-2 for more than 10 years now, so we know quite a bit about it. Still, we're not done, but we know we have a number of results on it. In normal conditions, first I want to say that most of our studies have been done in rats. And the reason why I've chosen rats from the beginning of my career is exactly for all those reasons that you have heard here today. They are more sophisticated, they are more complex, and I was interested in understanding how the physiology of the system changes with learning. I didn't want to manipulate the system genetically first to understand what that does. So I used rats for all those reasons. And we identified IGF-2 in rats, and we manipulated IGF-2 in rats. But then when it came to try and apply this in a therapeutic way, we thought that switching to mouse was a good idea, and it is a good idea for a number of reasons. One is that the mice are smaller, so we can actually do therapy in a less costly way. And 2, genetic models of diseases exist mostly in mice models, and only recently we're understanding that going back to rats is going to be important. So we started by looking at insulin-like growth factor 2 in the brain of rats and found that it is required for long-term memory formation. If we block the change of IGF-2 that happens with the learning, we block long-term memory. But then in other experiments, when we replaced the IGF-2 that we gave away in the control rats, we found memory enhancement. So we went to mice, and here I'm just summarizing the data in mice, and we gave actually an injection, subcutaneous injection of IGF-2 to these mice just before or just after an experience, a learning event. The advantage of IGF-2 is that it crosses the blood-brain barrier. And this is for treatment, is very important. We can actually now inject IGF-2 systemically in mice and look at what happens. And that's what we did. And in this slide, you see that if we inject IGF-2 in a mouse immediately before learning, we enhance the memory very significantly. So in blue are the control mice that receive vehicle injection, and in red, the mice that received IGF-2. And we see that the effect goes across many different types of memories, all mediated, all processed by the medial temporal lobe. Fear memories, which are memories for aversive experiences, recognition memories, recognizing an object, but also recognizing a conspecific, another mouse. And because of the effect on the social recognition memory, recognizing a conspecific, we thought maybe IGF-2 is going to be beneficial for potential therapies. So let's start to look at deficits of social recognition. And these are, as you very well know, typical of autism spectrum disorder. And in autism spectrum disorder, we have a number of symptoms: social impairment, repetitive and stereotype behavior, cognitive impairments. We used a mouse model that reproduces all these core phenotypes, and this mouse model is called BTBR. I can give you more information if you want later. But in these mice, when we inject IGF-2 before any of these deficits, when we challenge them, we repair them all, we reverse them all. One injection, subcutaneous, in the mouse. So we went back and said, okay, now we know IGF-2, this natural growth factor, promotes memory strengthening and persistent in physiological condition, is a potential therapeutic avenue for autism spectrum disorder and neurodevelopmental disorder. We also did other studies. We did it in aged rats who has cognitive impairments, and we found that the cognitive decay in aged rats is repaired. A colleague of ours tested the IGF-2 effect in Alzheimer's disease model in mice, and also 2 different labs reversed many of the problems that the Alzheimer's mice have. And so one question is, how? How does it happen that we see all these beneficial effects, many different types of diseases, many different conditions, and also we potentiate memories? I'm going to get back to this question in a little bit, because first, our next step was to find a genetic model of a human disease, and we decided to test Angelman syndrome. So I don't have to explain to you what the Angelman syndrome is. You will explain it to me every day and for a long time in the future, because I want to work with you. But what we did in the lab was to use the Angelman syndrome mouse model, which is the deletion of the UB3A gene in the maternal allele, and tested a number of phenotypes that these mice have. So the symptoms, as you know, include developmental delay, intellectual deficits, repetitive behavior, lack of speech, seizures, and motor problems. And we have tested so far with IGF-2 the ones that I cross in red: intellectual deficits, repetitive behaviors, and motor deficits. So I'm going to show you some data. This is, again, an injection of IGF-2 20 minutes before learning this fear conditioning experience. In this experience, the mouse is placed into a box, and they receive a mild food shock. And they learn that that context is associated with the shock, so next time we place them in, they're going to freeze if they remember the context. So this is a contextual memories, the memories that we're studying. It's a very classical test to look at this type of memories. So here, when we injected IGF-2 20 minutes before training and tested 24 hours later, this is the deficit we found. In blue, you see the normal mice. In orange, you see the Angelman mouse model in the performance of this memory. When we injected IGF-2, this is what we found. Memory enhancement in normal mice, recovery of the deficits in the Angelman mice. So, another type of memory, novel object recognition. This is non-aversive, it's just exploring objects and remembering what objects they have seen before. So the mice are habituated. A day later, they receive IGF-2 injection or vehicle injection, and 20 minutes later, they are exposed to 2 objects. So they learn about those 2 objects. And then 4 hours later, we test their memory. So we replace one object with a new one. And naturally, the mice explore the new object more than the old one, of course, if they remember it. And then 24 hours later, we test them again. We replace the new object with a new object again. So let's see the test at 4 hours. This is the deficits, normal mice, Angelman syndrome. This is the IGF-2 effect, complete repair of one injection of IGF-2. 24-hour test, both show memory enhancement. At this time, with this type of experience, the memory goes back to baseline. They don't remember, even the normal ones. But IGF-2 potentiates the memories. Spontaneous alternation is a task that measures exploration, working memory, the ability to remember what we are doing, and behavioral flexibility. This uses an apparatus with 3 arms. The animal is placed in the middle, and they start to explore the arms. And a normal mouse will explore the arms with alternation, A, B, C, A, B, C, A, B, C, because they remember that they visited A just before, so they go to B, and so on. But when there is repetitive behavior or lack of flexibility, the animals start to go back to the same arm or make the wrong ABC alternation. So if we look at the performance, which is the percent of the correct ABC choices, what you see is that the Angelman mice have a deficit compared to the normal, and this is the effect of IGF-2. We completely repair. Accelerating rotor rod. Here we are looking at motor coordination and balance. You have seen this already. I'm not going to explain it. We give IGF-2 or vehicle solution. This is the deficit we find in Angelman mice, again in orange compared to the blue, which are the normal mice injected with vehicle. And as well as the Angelman here, they're injected with vehicle solution. So this is the deficit. This is the effect of IGF-2. Again, full repair. Test 1 is done 20 minutes after the injection of IGF-2. Test 2 is done 2 days later, so there is no more injection here. The injection was done once. We're just looking at whether the effect is persistent. And 4 days later, again, is persistent. So there is a very persistent effect of IGF-2. Marble bearing. This task assesses motor ability and repetitive behavior. On a surface, we place marbles, and the animal starts digging and undigging these marbles. So we count the time that they spend doing that and the number of marbles that they bury and unbury. And this also shows an effect in the Angelman mice compared to normal. And this is the IGF-2 effect. Again, we reverse marble bearing. So how does it work, right? We see all these great effects. We really need to understand the mechanism. And this, a big part of the lab is actually doing that. So I cannot give you a lot of information, but I'm happy to give you a lot of information later. The reason is that we have so many studies ongoing, and the story is quite long, but I'm going to give you the essence of it. First, we know that this happens through the IGF-2 receptor, not the other receptors of the system. I didn't tell you, I'll tell you now, that IGF-1 and IGF-2 and insulin can bind the other receptors, even though they are not as strong, the binding between the peptide, the protein, and the receptor. They can. It's called with lower affinity, less strength, but they can. So they can activate other type of cellular responses. But of course, the strongest is with their own type of binding to the receptor. We know that if we block IGF-2 receptor, we block completely the effects of IGF-2. But if we block IGF-1 receptor, we don't block the effect. So all the effects we see are through the IGF-2 receptor. And that is important because IGF-2 receptor, what it does, it takes protein to lysosomal degradation. It has nothing to do with the other receptors that are implicated in growth. Okay, so the second point, and this is a lot of studies that we have ongoing in the lab, is that it targets mechanism of synapse and cell homeostasis because it targets the ability to clear the excess of proteins. It targets what is called macroautophagy. And macroautophagy is a way to get rid of the garbage in the cell. And so as Alison has mentioned at the beginning, UB3A, but many diseases, neurodegenerative diseases, neurodevelopmental disorders, they have an excess of protein that accumulates. And what IGF-2 seems to do is to clear the excess of accumulating proteins. And this is why we think it also works in aging, as well as in Alzheimer's disease and neurodegeneration. So if we have too much proteins, they're going to accumulate, they're going to start being deficient in functioning, and over time they can degenerate, or they can remain impaired for quite a long time. So IGF-2 cleans the cells from the garbage. So specificity. We want to identify something that is more specific for the IGF-2 receptor that does not bind to the other ones. And so we identified one novel ligand to the IGF-2 receptor. I'm going to call it L1. And we have started to look at the effects, the behavioral effects. And here I'm going to show you 2 slides about L1 injected, again, systemically, subcutaneously, in mice. This is the novel object recognition that by now you know. This is the deficit of the Angelman mice compared to controls injected with vehicle. This is the injection of L1. Again, L1 brings the level of ability to object recognition to the level of the normal mice. And the normal mice with L1 have increased ability to process the information, increased memory for it. This is at 24 hours. In both cases, like IGF-2, we see memory enhancement. This is a new task that I didn't explain in IGF-2, the hind limb clasping behavior. And I think Jill actually showed that, the hind limb, sorry. And this clasping behavior is typical of motor deficits, motor impairments. So it is shown in many different types of models of motor deficits. I'm going to go because I have 1 minute. As you can see, the hind limb clasping behavior is significantly higher in Angelman mice, and this is what L1 does. It reduces these motor deficits significantly and very close to the normal mouse. So in summary, IGF-2 is a potent memory enhancer that reverses cognitive deficits, social interaction impairments, and repetitive behavior in models of autism spectrum disorder. It reverses cognitive deficits, repetitive behavior, and motor impairments in a mouse model of Angelman syndrome. Selectively acts via IGF-2 receptor, and IGF-2 receptor ligands, other ligands have similar effects. The mechanism of action target protein engulfment and synaptic dysregulation in a variety of synaptic disorders. So we also we aim at progressing the studies and as soon as possible toward clinical translation. And I want to end by thanking who actually did the work, Janina Descalzi and Adam Steinmetz, two postdoctoral fellows in the lab. Adam has left the lab for a position, and Janina is leaving soon for her own position. So I'm actually looking for someone. If you know of someone interested in continuing these studies, direct them to me. I want to thank former lab members, Sarah Stern and Amy Coates, who did a lot of work on the autism model, and of course, the funding agency, in particularly FAST, who really believed in these studies and funded all the behavioral studies that I've shown you in the Angelman syndrome mouse model. We really had only some preliminary data, and this is where we are now. And this is the current lab, and I want to thank the collaborators, and last but not least, the FAST Consortium research team. I'm collaborating with Ed Weber, Ann Anderson, and Jill Silverman to move this to their beautiful rat and models. Thank you very much for your attention.
Please feel free to ask a couple questions to Dr. Alberini.
Is there any diabetic implications with the therapy?
Yeah, very, very good question, because being part of this system, one think, oh, what if? Actually, not that we have seen. We have tested a number of parameters, but also not from the literature, because IGF-2, through the IGF-2 receptor, doesn't involve the insulin system at all. Yeah.
So, quick question.
Yes, here.
Yeah, great talk. That's beautiful work. So, Alzheimer's disease is more of a proteinopathy, and Angelman syndrome is not. So, what do you think the molecular mechanism of action is with the IGF-2?
Yeah, of course, that's $1 million question. We are working on that. And so our data indicate that IGF-2 promotes autophagy. Autophagy is linked to the regulation of the mTOR pathway. And in Alzheimer's disease, there is an accumulation of proteins, and the accumulation of proteins is not clear, to the point that then there is, amyloid deposition and so on and so forth. In addition to that, I must say that amyloid deposits may not be really the answer to all the forms of Alzheimer's disease. So we don't know what the sporadic form of Alzheimer's disease actually are in terms of their etiology and mechanisms. So we know what we know from the genetic models, and also, of course, from human description of the pathology, but we really don't know what are the mechanisms that in the sporadic form of Alzheimer's disease get impaired to lead to neurodegeneration. However, we know by and large that they have to do with protein processing, protein accumulation. For example, amyloidosis is one, right? And autophagy is strongly impaired in Alzheimer's disease. So that is the link between autophagy, IGF-2, and Alzheimer's disease. In Angelman syndrome, that's not the case. However, UBE3A is regulating ubiquitination, which is important for degradation of proteins and for the accumulation also, when it is impaired, of aggregates. Now, the crosstalk between ubiquitination and macroautophagy is not clear, but papers are suggesting that there is crosstalk. So this is where we want to get the understanding through the IGF-2. What is going on? What is wrong in the Angelman syndrome? What is wrong in the Alzheimer's disease? And how does IGF-2 repair that? Long story short to say, we are working on it. Yeah.
Over here. One question here. Hi, great presentation. Thank you very much. I have a question. Is there a link between the loss of the function of UB3A and a loss of IGF-2? Is there a link?
We
Do
don't
we
know.
know that?
We don't know. We are studying that. We are looking at that. We don't know. We don't know yet. The regulation of the levels of IGF-2 are not normal in the Angelman syndrome mice.
Okay.
But we have to study it in humans. We are at the very beginning. So I cannot answer the question. In the mouse, they are not normal. But the studies we have so far is in the mouse without even being challenged with experience. I think the biggest changes we're going to see, the biggest biological markers we're going to find is when we challenge them with experience, let's say a learning experience. And we have this in progress.
Okay, thank you very much.
Thank you.
Wonderful. Thank you so much.